going to do is sort of combine both the lectures because I'm going to go back and forth between the stem cells, the cancer stem cells, and the cancer cells. So what I would do is, I divided this talk into 3 separate sections. One we would deal with understanding what the stem cells are, then, understand the role of non-GAGs, which is the N-glycan, and then the glycosaminoglycans in regulation of cancer stem cells, or cancer cells. All right, so this is the learning objective for the first portion of the talk. So what we want to do is understand what the stem cells are and able to distinguish the embryonic stem cells from the induced pluripotent stem cells and the cancer stem cells, because these are the terms that you would come across quite often if you do the search using stem cells either in Google or in PubMed. All right, so then we would focus mostly on, the specific examples from literature, looking at the role of N-glycans in either identification or regulation of cancer stem cells, and, understand the application of doing research with N-glycans and how, that can be used in the translational setting. So those are the objectives. The stem cell theory, briefly, there are 3 terms that you'd come across: the embryonic stem cells, the induced pluripotent stem cells, which are essentially similar. They are the cells which can give rise to a lot of different tissues and would be, would have a lot of application in regenerative medicine. Whereas the cancer stem cell is a very specific term that's only used in the cancer literature. And this term recognizes cells that behave like stem cells and have certain properties that make them treatment resistant and hence require special attention. All right. So the stem cells essentially are biologically speaking cells that possess the ability to differentiate as well as self-renew. Those are the 2 important characteristics of a stem cell. In other words, the stem cell has to make a copy of itself, which is self-renewal, as well as differentiate into many different types of cells, so it can make a functional tissue where if it's a normal stem cell, or make a lot more cells which are, which can form the bulk of the tumor. Okay. So they're found in the embryo as well as the adult tissue in the multicellular organism. And when we talk about these stem cells, we would also use the adjectives to define them based on their potency. So they can be totipotent. In other words, they can give rise to not only the embryo, in other words, the baby, but also the tissue that helps the baby obtain nutrition from the mother, like placenta, etc., etc. So those are the totipotent stem cells. The pluripotents are the stem cells which give rise to all tissue types in that particular organism. So if it's human, all 3 tissue types, which is epiderm, endoderm, and mesoderm, can be formed from a pluripotent stem cell. Whereas multipotent are, more differentiated. The example would be mesenchymal stem cells, where, one mesenchymal stem cell can make different types of cells, for example, immune cells or fibroblasts and things like that. But they can't make other epithelial or endothelial cells. So they have somewhat differentiated, whereas unipotent ones can only give rise to one cell type, essentially. So as we go down the hierarchy, there is more differentiation and more commitment, essentially. Okay, so quickly through some of the other things, the embryonic stem cells traditionally used to be derived from this embryo, the blastocyst stage, and then those cells were reimplanted into another mice to form teratoma, from which you can derive, bunch of endothelial cell culture. And now this was a bit of a controversial approach. So, people started looking into different things by which the differentiated cells can be turned back into pluripotent stem cells, which is what we call induced pluripotency. In other words, you take the more differentiated or unipotent cell and make it pluripotent. And for that, we needed to understand what are the factors that regulate differentiation from pluripotent to unipotent, as well as in the other direction, if those unipotent cells were more plastic, so to speak. So the Nobel Prize winning research This research was awarded Nobel Prize not too long ago in 2012. People from the investigators from Japan and Caltech collaborated and showed for the first time that you can indeed take a skin cell or a bone marrow cell and reprogram it to turn it into an induced pluripotent stem cell, which is a functional equivalent of embryonic stem cell. And hence, it can be used for all the research that you need to do with embryonic stem cells. And this is basically a schematic showing that certain factors were involved, which had to be identified. And of the 4 factors that they ended up using, 3 of them are shown here to show how sequentially they can turn the terminally differentiated cell into an intermediate state, which is a prime cell, and then into induced pluripotent stem cell. So what they showed was using these 4 core transcription factors, and you'll have to know this 4 core transcription factors: OCT4, SOX2, KLF4, and MYC. Of this, MYC is actually an oncogene. In other words, if it's overexpressed in cells, it can actually make them cancerous. But using the combination of these 4 factors, using viral vectors to transduce this 4 transcription factor into a more differentiated cells, they were able to obtain colonies of these, induced pluripotent stem cells. But you look at this number, the reprogramming efficiency was fairly low. In other words, you are successful in only about less than 1% of the cells. You can actually induce pluripotency. And also it's a bit tedious because you have to have this feeder cell layer on which this growth has to happen and things like that. All right. So changing gears, now this is a second concept. We talked about the embryonic stem cells and the induced pluripotent stem cells. Essentially, those are functional equivalents and would be used in regenerative medicine because you can use these cells to make any tissue. In other words, if there is injury, you can inject these cells, and hopefully, if you have the right plan, you can differentiate them into, wherever they're being injected. If it's a nervous tissue, the neuronal cells can be, made out of them, and you can help people recover who had spinal trauma, et cetera. All right. So what about that, applying that concept in cancer? So, as I said, all the adult organs have this unipotent stem cell, and in certain organs they're very active, like intestine, hair follicles, and bone marrow. You continuously need to regenerate your cells, so in order to, because most of the cells are made by these organs are continuously lost. So the stem cells are fairly active. They have to not only keep their own population going, but at the same time make all these other functional cells. And so in these organs, the stem cells, you can easily obtain and culture ex vivo. So in the intestine, they lie at the base of the crypt, and, there's one stem cell shown here, which basically, if it acquires certain type of mutation that makes it more cancerous, then it retains all its stem-like properties, but becomes a cancer cell. And so it would be called a cancer stem cell. And that cancer stem cell would have the same property, which is to self-renew, in other words, making a copy of itself as well as differentiate, giving rise to, bulk tumor cells, which would eventually give rise to cancer. And this process of normal stem cell differentiation and self-renewal is regulated by some of the growth factors and morphogens, which are targets of glycosaminoglycans as well as other glycan molecules. So the cancer stem cell theory, this translational application is that the cancer, if you kill the cancer cells using the standard chemotherapy, they mostly kill the non-cancer stem cells, and the cancer stem cells survive and then give rise to the tumor, causing relapse. But if you take care of the cancer stem cells, then, tumor eventually would lose its ability to regenerate, and this would eventually disappear. It would be a slow process, but you can achieve complete remission, essentially. So, this is just to show that these cancer stem cells can actually be studied in vitro using a variety of methods. The most common methods that are used are growing them as spheroids, which is shown in that top left panel, or through the flow cytometry by identifying certain markers on these cells that they express more often, or using this site population assay wherein you're trying to block a transporter of a dye, which is then retained, and it preferentially is retained in the cancer stem-like cells, basically because they have very high amount of this transporter called ABCG2 transporter. So these are the variety of methods that are used to study cancer stem cells and in vivo. This is not important, so I'll skip over it, but essentially what I was showing in this slide is that, you can use some surface markers to identify these cancer stem cells. And the most important point is that most of these cancer stem cell markers that we would review in the next few slides are actually glycoproteins. And it's just the conversion from PowerPoint to PDF has caused loss of some of the features. So I'll be happy to share the PowerPoint slides with you guys. Okay. So what is finally, once we reviewed those two concepts, the induced pluripotent stem cells as well as the embryonic stem cells, which are useful for regenerative medicine, as well as cancer stem cells, which are useful for better anti-cancer treatment development, is there a relationship between the two? Indeed, there appears to be some relationship because the induced pluripotent stem cells, you could potentially convert them into cancer stem cells. And not only that, it has been shown that cancer stem cells can actually be reprogrammed into induced pluripotent stem cells, in other words, to allow them to function more normally and lose some of their cancer-causing properties. So, it turns out that, if you take the mouse embryonic fibroblast and, induce them with the four pluripotent factors that I described before, you can get the induced pluripotent stem cells. But on the other hand, if you change the composition such that, you have, cancer-causing transcription factors, mostly MYC transcription factor, as I mentioned before, along with some of the other ones that help promote cancer growth, then you can convert them into these cancer-like cells. But when you put back the core transcription factors minus the MYC, they actually get converted into more normal, obviously the cells with more growth potential, but more normal like cells. In other words, more benign type of growth rather than malignant growth. Okay. And all of these processes are very complex and are regulated at the level of the chromatin. It usually involves ultimate regulation of the stem cell involves the epigenetic processes. Okay. And these epigenetic processes are controlled at the cell surface through these different receptors which signal into the nucleus. And hence, you can potentially regulate that stemness through these growth factor cytokine morphogen receptors, essentially. And the reason why we are interested in studying them is because most of these receptors and or their ligands bind to glycosaminoglycans or other glycans. And that's how the glycans regulate cell growth, especially cancer growth and cancer stem cell growth, as well as normal stem cell growth. Okay, so looking at some specific examples, we'll start with the N-glycans, and, since you guys have already done some of the lectures on structural aspects of the glycans, we won't go into the details of this, but, essentially, the N-glycans as well as the glycosaminoglycans are the major type of glycans. And in cancer specifically, if you look at the structures that are more commonly altered, the glycan sequences that are more commonly altered, you would find that both the GAGs expression as well as the N-glycan levels and the sequences are often aberrantly expressed on cancer cells compared to their normal counterparts. And so that's why, one probably should study to understand what role they play in regulation of the growth of these cells. So these are various markers that have been looked at on cancer stem cells as well as the induced pluripotent stem cells. And many of them, you can recognize they're all types of glycan molecules, mostly N-glycan, because, for this section, we're focusing mostly on the N-glycans. And so you can see a variety of changes like fucosylation, Sialyl acid related changes, and many other common antigens that we know of, the glycan antigens, are aberrantly expressed in either the cancer stem cells or the induced pluripotent and the embryonic stem cells. Okay, so looking at some very specific examples, CD133 is a very common cancer stem cell marker. It is found in almost all cancer stem cells. In other words, from the brain all the way down to intestine, most of the cancer stem cells express the CD133 molecule. And what this study shows is that if you have the appropriate N-glycan modification on the CD133 molecule, then it's recognizable on the cell surface. But if you do lose that, which is on the rightmost panel, then you don't express CD133, or at least it's not in the detectable form. At least not in the detectable amount using this particular antibody. But it's also been looked at with other antibodies too. So it certainly affects probably the, if not the synthesis, at least the presentation of the receptor on the cell surface. All right. And other groups have done more intense profiling of N-glycan in the serum as well as on the tumor cells, and to try and come up with some circulating biomarkers. And what they have found is that there's most of the abnormalities seen in cancer fall into three major categories. One that relates with the branched N-glycan, fucosylation, as well as sialylation. And so those are the common abnormalities I've seen on the glycome, found in the glycome of the cancer cells. So they looked at specific abnormal changes in the branched N-glycan, especially the MAN1A1. You can see some of the enzymes related to the branched N-glycan synthesis are abnormally expressed in the CD133 positive, which are the stem cells, compared to their non-stem-like counterparts. Now, the expression is not that high in this particular, it's not that altered in this particular case, but this is just to give you an example. There are many other examples where when you do better sorting for the cancer stem cells, not using just one marker, but maybe few other markers, a complement of markers, you might be able to identify changes which are significantly altered between the stem cells and the non-stem cells. All right, so, some folks do the glycomic analysis, then the basis of those glycome alterations are, can be pinned down to the enzymes that are responsible for introducing certain glycan modifications. So, one can hypothesize that certain enzymes involved in N-glycan synthesis, and, by extension, maybe the glycan degradation or, things like that, could be altered in cancer or cancer stem cells. And one most commonly studied enzyme is Sialyl transferase, ST6-GAL1, in the induction of hypoxic responses. So basically what this study looked at is in pancreatic cancer cells, when there is hypoxia, there is an induction of the particular enzyme, and hypoxia is commonly associated with poor outcomes in cancer, like invasive growth, as well as induction of cancer stem-like cells. So one can hypothesize that this enzyme might play a role in the aggressive behavior of cancer. The other example is Sialyl Lewis X antigen, which is one of the well-studied glycan antigen. It is actually a marker of squamous cell CSCs. In other words, the cells expressing high level of Sialyl Lewis X behave more like stem cells. And, there are some examples shown here using flow cytometry. If you sort these cells, you can find higher expression of, so if you sort for the CSC markers, then you can find higher expression of the enzymes responsible for synthesis of this particular sequence. And also at the functional level, you can see increased formation of the spheroid, which is one of the technique I showed you earlier to study. Spheroid formation is one of the techniques to study the cancer stem cell growth in vitro ex vivo. And, if you use these, if you use the inhibitors of this enzyme, you can see a clear difference in the spheroid formation, suggesting that it might affect the function of, it might be functionally relevant in regulation of CSC growth. Okay. And again, in another cancer, Sialyl Lewis expression in the circulation, in the circulating cell, was seen to be higher in the cancer, cells obtained from the cancer patients than in the normal healthy control, suggesting that the cancer cells might, in general, increase expression of this particular antigen on their cells. All right, so, essentially you can use this concept and apply this to discover novel biomarkers where you can take tumor samples as well as matched plasma from the patient and then analyze the glycom, the N-glycan, to try and come up with the markers. And you can be more selective and look at specific markers in the tumor on the CSCs, which is the cancer stem cells, which can distinguish them from non-cancer stem cells and try and figure out if those markers are indeed seen in the plasma. And, depending on the level of the expression of that antigen that distinguishes CSC from non-CSC, you could potentially theoretically develop a biomarker of CSCs, which is glycan-based. By the way, most of the cancer biomarkers are actually glycan-based biomarkers. You might know that. Okay, so another another technique can be using lectins, to isolate CSCs. Lectins can bind to, some very specific sequences, and you can use them as probes to detect certain sequences, and then can be used, as a probe for CSCs, just like we use antibodies nowadays. You can develop lectin-based assays for cancer stem cell detection. All right. Now, what about so this was all about the cancer stem cells, but also what about the normal stem cells? Some of these enzymes that we discussed before, like FGFR1, we observed that it was significantly increased in the cancer cells, but it appears that, if we look at the induced pluripotent stem cells, that's panel D, or the left-hand figure panel, there's panel D in that. You can see a clear overexpression of FGFR1 in induced pluripotent stem cells. So, this enzyme, which actually control the levels of specific glycan sequences, could be significantly altered in stem cells, and as we saw before, they might actually have a functional role. In other words, altering the levels of this enzyme can significantly impact the stemness, both the cancer stem cell property, or even the normal stem cell stemness property can be influenced by this molecule, by this enzyme, which regulates certain glycan sequences. So again, as I said, of the three major aberrant sequences, the branched N-glycans, the Sialyl acid modifications of N-glycans, as well as the fucosylated glycans. This is an example where branched N-glycans actually play a role in regulation of the cancer stem cells. So this is another enzyme. GNTV expression was seen to be high in cancer stem cells, and then by modulating the levels of that, you can see inhibition of the in vivo spheroid growth as well as the tumor growth in the animal. So, and this is again a different kind of mouse model where this is a model of cancer initiation. In other words, this is early stage cancer. Like this hasn't become an invasive cancer, but it's in the polyp stage. So intestinal cancer, the colon cancer develops from normal mucosa. You initially develop polyps, and if they're left alone, they can become cancer. So this, the branched N-glycans may actually regulate CSCs even in the early stage of cancer development before it becomes invasive, because you can certainly influence the development of this colonic polyp in this ATC mean mice model with the GNTV knockdown. So again, suggesting that, it might play an important role in cancer stem cell regulation very early in the cancer development as well as the cancer progression. Okay, so another enzyme, B for Galnac 3 for the Lacdinac modification. It's again, it shows similar changes, essentially it acts as an oncogene, so to speak, because the overexpression, as you can see, leads to poor survival because it's overexpressed in these cancer stem-like cells, and its expression is associated with poor survival, suggesting that it might also play a role in regulation of cancer stem cell growth. And again, similar biochemical experiments to modulate the levels of the enzyme significantly affect the growth of the cells, as well as the cell intracellular signaling with respect to growth factors. So this is the first, experimental evidence of the hypothesis that we had raised at the beginning of the lecture, is that the reason why we should study N-glycans or glycosaminoglycans is that they can significantly influence the surface growth factor morphogenetic cytokine function, which in turn regulate, many intracellular signaling cascades and eventually the stemness. Okay, so this is one example, is that the epidermal growth factor receptor, it's signaling, not its level, but it's signaling, meaning the phosphorylated form of that molecule is significantly inhibited. Okay, so again, there's just a few more examples of this. Again, we're back to CD133 molecules. We had discussed that if you don't have N-glycan modification on this molecule, its expression on the cell surface is lost. And this study actually looked at more specific, sites of N-glycan modifications and its influence on its expression as well as its role in regulation of cancer stem cell growth. And it certainly appears that, the N-glycan modifications are important for both the expression of the molecule as well as its functional role in regulation of the cancer stem cell growth. Okay. Now, the other area which is very hot in the cancer therapy is the development of immunotherapy, which has found indication in variety of cancers. I would say almost 75 to 80% of the cancers now, immunotherapy plays a major role, and no cancer patient dies without seeing immunotherapy. And in certain cancers, it provides very prolonged responses leading to cure, which, was difficult to achieve with the traditional treatment that we had for cancer. And this is a study that was published in Cancer Cells showing the importance of glycan modification of the molecules against which these targeted therapies are directed at. And these are the molecules called checkpoint molecules. In other words, what they do is when these checkpoint molecules are expressed on the cell surface, especially if they're negative or the inhibitory checkpoint molecule, even if you have the immune cells recognize the cancer cells, they can't kill those cancer cells because these checkpoint molecules put a brake on the activation of those immune cells. So they can be there but not activated. So these molecules are significantly modified by the glycan, and that glycan modification of both the checkpoint molecule as well as, in this case, the antibodies are directed against the against this checkpoint molecule. If they are modified by glycans, then their potency as well as the efficacy significantly improves by specific glycan modification. So in other words, you can regulate the function of those checkpoint proteins themselves, as well as the antibodies that are developed against those proteins can also be glycan modified to improve their efficacy. So in both cases, there is a potential translational role of glycans, especially in cancer therapy, cancer immunotherapy. I think I gave you several examples of enzymes that regulate N-glycan synthesis and their role in cancer. These are some of the examples of what the O-glycan regulating, the O-glycan sequence regulating molecules have in cancer stem cell growth regulation. Similarly, just like GNTV and some of the other enzymes that we looked at, for example, ST6-GAL1, the O-glycan specific enzymes involved in O-glycan synthesis also can act as oncogenes. And this is shown in this example, as well as its role in cancer cell signaling. So again, I'm, looping back. Initially, we started with that hypothesis is the reason why we should study glycans is because they regulate this signaling that is critical for cancer growth. And this is another example where aberrant O-glycan synthesis promotes this cancer metastasis through specific growth factor receptor signaling. And in this case, it's the FGF receptor. Okay. So again, all these examples are showing you that both N and O-glycan sequences there is aberrant expression of certain N and O-glycan sequences, which can be traced back to the enzymes that regulate their expression. And by targeting those enzymes, you can alter the expression of those sequences as well as affect the function of the cells in which they're aberrantly expressed. And one of the mechanisms seems to be through the regulation of growth factor, cytokine, morphogen receptor signaling at the cell surface. Okay? All right. So what is the implication of the research, both the translational research as well as the basic research implication of this certain specific examples that we looked at? One of the things that, one of the concept that is emerging is this integrated glycomics and using that multidisciplinary approach to characterize the structure, function, and the regulatory role of aberrant glycosylation in not only cancer but any other disease. But in this case, we're just going to focus on CSCs, or the cancer stem cells and other stem cells, and how this can be applied. Okay, so one can imagine that you can do multi-omics, and nowadays it's very easy to do multi-omics approach on a selected cell population and trying to integrate all the information. So you start with the glycome, the glycomics, as well as the transcriptomics and or metabolomics, the proteomics, whatever your omics interest is, and then try and combine those information, try and integrate the information to come up with the regulators of these aberrant glycan structures, whether that's an enzyme, whether it's a microRNA that is responsible for regulating enzymes or the other proteins, et cetera, and then by deciphering the specific targetable changes in the cell that might be regulating the abnormal glycan structures, you could devise treatment against those targets, and also study the function of that specific glycan molecule by altering its regulators, essentially. And you can do both the functional studies as well as develop novel therapeutics. Okay, so this is, I think, the first portion of the talk. Went through those slides a little quickly because I wanted to spare some time after this section before we jump to the next section for the questions and answers, because I'm sure there are certain areas that we would need to revisit. But in conclusion, I would say that the most important thing that we learned from some of these examples is that stem cells, whether they are, normal stem cells, which are the adult stem cells, or in certain cases induced pluripotent stem cells, but mostly cancer stem cells, they show differential expression of certain N-glycan and or O-glycan sequences, which can be used as potential biomarkers, because we showed, we looked at some of the approaches, like you can use lectin array approach, you can do serum and glycomics, and try and come up with biomarkers to identify the proportion of the cells in a given system, basically, in a given biological system. And also you can use these markers to isolate specific cell population too. So we showed certain examples of, cancer stem cells, can be isolated using these glycomic markers, essentially, which is not done. Mostly we, isolate them using the complement of protein markers, but, all these proteins are modified by N-glycan. And so there certainly is a value in doing research along this line where instead of the protein level, it might be the, specific modification, the glycan modification on the protein, which might be a better marker of the stem cell. And not only that, not only are they, differentially expressed, so we're not just looking at the, their utility from a biomarker perspective, but we have also looked at certain examples, specific examples, where we have shown that these altered sequences actually play a functional role in regulation of, in the case of cancer, the cancer stem cell growth, or in case of the other stem cells, the induced pluripotent stem cell growth can potentially be regulated. And the third most important point is that we can use very powerful omics, techniques, glycomics, and next-gen sequencing, as well as the other omics techniques, and do integrated glycomics to not only develop novel biomarkers, but also to understand structure-based, functional roles of these glycan sequences, as well as identify certain cellular processes that regulate the expression of these abnormal glycan sequences, so that we can identify novel targets for the therapeutic uses. Okay? All right. So before I go to the next section on GAGs, why don't we just pause for about 5-10 minutes, and if you guys have any questions on the things that we covered so far, we can go over that. Just to recap, so that we don't forget, what we did so far is to understand the differences between the embryonic stem cells, the induced pluripotent stem cells, and the cancer stem cells, and the fact that the 4 core transcription factors, the OX4, SOX2, NANOG, and CMYC, can be used to turn the differentiated cell into a pluripotent stem cell. In other words, suggesting that none of the cells in the body, if appropriately modulated, have lost their plasticity. In other words, even the most differentiated cells that we think are terminally differentiated can be reprogrammed into this stem-like cells, and that concept is the pluripotent, the induced pluripotency, and the cells that you get out of that are induced pluripotent stem cells. And that's the Nobel Prize winning research, obviously, because, it circumvents a lot of different issues that were ethical and, logistic issues that were involved with doing research with the embryonic stem cells. Okay? So that's the normal stem cells for regenerative medicine. And then we also understood how those normal stem cells can get converted into cancer stem cells and retain the stem-like properties. And they're really the cells to go after in cancer, because they're the ones who resist killing by the standard cancer treatment and then prompt relapses, which are the major reason why most of the cancer therapies are not able to achieve long-term remission and or cure. And we saw that both these cancer stem cells as well as the normal stem cell function can potentially be regulated by certain N-glycan sequences, and those sequences, which the synthesis of those sequences is regulated by the N-glycan synthetic enzyme, which can serve as potential targets for cancer treatment and or regulation of the normal stem cells, in which this specific sequence and or their upstream enzymes are dysregulated. And using that same concept, extending that, you could apply that into, developing these new ideas along the integrated glycomics theme to develop, to conduct more unsupervised analysis to identify functional regulators, so identify abnormal glycan sequences as potential biomarkers and identify the regulators, more upstream regulators. Okay, so continuing the same thing, now that we understand the stem cells, the cancer stem cells, and have looked at the role of the N-glycan, I would now focus on glycosaminoglycans because these molecules are of high interest to us. We work with these glycosaminoglycan sequences, the soluble glycosaminoglycan sequences, to try and modulate the cancer stem cell function in vitro and in vivo. And as you might know, the glycans can be broadly classified into both the sulfated glycans, the sulfated as well as the non-sulfated glycans. And the sulfated glycans are heparan sulfate, chondroitin sulfate, and dermatan sulfate, as well as keratin sulfate. But the non-sulfated glycans are also just as important in cancer, and they are the hyaluronan molecules. And these hyaluronan molecules are present in the stroma of the cancer, where they serve two different functions. One is obviously the traditional function attributed to the GAGs, is to interact with certain proteins, and we would look at specific examples. But hyaluronan is so abundant in the matrix that it also has some physical properties which has impact on cancer growth. And once the hyaluronidase, the pegylated hyaluronidase, is being tested in cancer to essentially alter that physical property of hyaluronan, which leads to increased pressure in the interstitium, so that the blood supply to the cancer is significantly affected. And then by removing this hyaluronan, reducing the interstitial pressure, you can reestablish the blood flow and the delivery of the cancer anti-cancer treatment. We're not going to focus on those physical aspects of the glycosaminoglycan's role in cancer, but we're going to focus more on its sort of biological properties that involve its interaction with certain proteins, especially the surface growth factor, cytokine morphogen receptors, through which they regulate the function of functions such as cell growth, migration, invasion, etc., and also the stemness property of the cell. Okay.
So, Bhaumik, did you finish the first lecture?
Yeah.
Okay. Can we take a 5-minute break?
Yeah, absolutely. Yeah. Why don't we take a 5-minute break, and then I'll. 46 is the learning objectives that we were laying down, the learning objectives for this portion of the talk. As I said, the focus of this would be to understand the role of both sulfated and non-sulfated glycosaminoglycans in the stem cell properties, for the normal and the cancer stem cell properties, and applying certain principles for the translational research, especially the therapeutic application of this, in targeting the cancer stem cells, is what we would discuss briefly. Excuse me. All right. So what are the physiological functions of the GAG? This slide essentially captures some of those functions that we attribute to GAG, which all can potentially, some or all can potentially play a very important role in regulation of cellular growth, especially cancer stem cells and the cancer cell growth. So, what do glycosaminoglycans do? So, glycosaminoglycans are both on the cell surface, anchored via the proteoglycans, but also in the matrix. And in the matrix, in other words, outside the cell, they actually serve as depot for a variety of growth factor cytokine morphogen molecules. And so if this is disrupted, all of a sudden you can imagine that, these molecules will be released. And, this can potentially be done through the heparanase molecule, which can cleave these glycosaminoglycan sequences. Okay. The other is that, these molecules on the cell surface, this glycan, the GAG chains on the proteoglycan can essentially bind to the ligand and or receptor complexes on the same cell or in the trans fashion on the other cells and regulate the activation of those molecules. The other role of glycosaminoglycan can be they can serve as receptors for certain molecules. You know about the viruses use glycosaminoglycan for entry. More recent evidence suggests that vesicles, so these are the exosomes. You might have heard of exosomes. These are vesicles made by the cells which carry many functional entities, including DNA, RNA, proteins. They use glycosaminoglycans as potential receptors for internalization into the target cell. So that could be a potential function of the glycosaminoglycan. So you can imagine that glycosaminoglycans can play a variety of roles in a given context, and one of that could be more important than the other, depending on the context. Okay. All right. So what are those specific, just to summarize what we saw in that picture, is that the glycosaminoglycans can serve as depots for all these ligands. They can also act as receptors for morphogens and create a morphogen gradient, which essentially we didn't talk about, but the morphogen gradient is what we alluded to in the initial slide when we showed that the cancer stem cells in the intestine lies at the bottom of the crypt and not only is responsible for maintaining its own population, but giving rise to all the cells up the top of the crypt. And in order to do that, you have to have a specific ratio of morphogens that promote self-renewal versus that promotes more differentiation. And so in order to maintain that specific ratio, in order to maintain that homeostasis, you have to have that morphogen gradient. In other words, as you go up the crypt, you have less and less of Wnt signaling, but more and more of PMP signaling. That is achieved through glycosaminoglycans. Okay, so the other function that we looked at was that they can act as co-receptors, because what they can do is bring the ligand and the receptor together or keep them apart and affect the signaling in the cell. And the last and not the least important is the endocytic processes, both for internalization of viruses as well as, we said, the important vesicles like exosomes, which can regulate the growth of cells distant from where they originated from. And exosome research is becoming very hot nowadays. And you can imagine that, one of the targets of interrupting that exosome-driven cell growth could be through the glycosaminoglycan. Okay, so these are the various enzymes. You've already studied them. They're involved in GAG synthesis. And we would look at the specific examples to elucidate the role of glycosaminoglycans in both the normal developmental processes as well as the cancer. And so, if a molecule plays an important role in normal stem cells, disruption of the enzymes that are responsible for the synthesis of that molecule, should significantly affect the developmental processes. In other words, if you generate embryos with deficiency of one of these enzymes, either it could be fatal, in other words, the embryo won't develop, or that, depending on the function of the enzyme and the sequence that, the abnormal sequence that results because of that, you can have a viable embryo which does not develop normally. So these are called developmental defects. Okay. So there have been experiments that have been done examining, just knocking out each of these enzymes, and it's a very standard biochemical approach to study the function of a given enzyme or a protein, is to generate knock-ins or knockouts. Okay. So we'll look at specific examples of some of the knock-ins and knockouts. All right. In this table, I have summarized some of those key experiments that have been done with the knock-ins and knockouts of specific proteoglycans, which are the proteins that carry glycosaminoglycan chains on the surface of a cell, or the enzymes that are involved in the synthesis of glycosaminoglycan chains, either synthesis and essentially elongation of the glycosaminoglycan chain. All right. So some specific examples, for example, glycans are the proteoglycans. Like syndicans, the glycans are very important proteoglycans, and they can be altered not only in cancer, but may also be upregulated in certain types of stem cells. And it turns out that when you have this knockout of glycan 3, you get very specific abnormalities in certain stem cell signaling, which deals with hedgehog or Wnt beta-catenin. If you guys get into the stem cell research, the most common morphogens that you would encounter would be the hedgehog pathway, the notch pathway, as well as the Wnt beta-catenin pathway. These are the three most important developmental pathways which regulate stemness, as well as some of the growth factors, including insulin-like growth factor and its signaling, as well as the epidermal growth factor and the FGF, which is the fibroblast growth factor, and its receptors. Okay, so you would see in the table that many of these critical players that regulate stemness seem to be altered. I mean, their signaling seems to be altered if you do the knock-ins and knockout type experiments with the proteoglycans or the GAG synthetic enzyme. So glycan 3 produces a specific syndrome where there is dysregulated growth. Essentially, you have too much growth because there is too much of the hedgehog and Wnt signaling when glycan 3 is lost. When glycan 6 is lost, there seems to be a specific syndrome called homodysplasia, where you have some abnormal growth, which is again blamed on the abnormal morphogen signaling as a result of glycan 6 loss. Okay. If you do more fundamental, if we get into the fundamentals, like, remove the exostocin function, it versus just the partial knockdown, it behaves very differently. So exostocin knockout, as you would imagine, because it's a very critical enzyme for the GAG synthesis, because essentially you have no GAG chains if you remove exostocin. There is a promotion of embryonic stem cell self-renewal. In other words, stem cells can grow better, and this has to do with the FGF signaling. In other words, what FGF signaling does is, it might sound counterintuitive, but the FGF signaling, even though it's a growth signaling, when it's activated in the stem cells, it acts as a differentiation signal. So the self-renewal and differentiation are counterbalancing, reactions in a cell. So when there is increased self-renewal, there is less tendency for differentiation, whereas when there is more differentiation, there is less self-renewal. So when the FGF signaling is not so active, you get this self-renewal in high gear, and it goes up. But when you have the partial knockdown of exostosin, it seems to produce the phenotype which is somewhat different. It's actually, there is less of self-renewal because there is increased FGF signaling. And it turns out it's because of the exostosin knockdown, there is compensatory changes which lead to altered sulfation of the GAG chains, and which might be responsible for some of these changes that are seen with the FGF signaling. So it depends not only on the type of deficiency, but also the degree to which that particular enzyme is absent, regulates the phenotype in a very specific manner. All right. So what about the N deacetylation and sulfation process? So NDST1 and 2 knockout, there is impaired embryonic stem cell differentiation, because it turns out that not just FGF, I've not listed here, but there is now emerging evidence that BMP signaling is critically regulated by N-sulfation. So if you don't have N-sulfation, you can have significant abnormalities in FGF signaling and BMP signaling, which can affect development of specific organs, especially lungs. Lung development can be impaired. All right. So then, what about the O-sulfur transferases? So the 3-O-sulfur transferase is a very important enzyme, which has shown to regulate the cancer stem cell self-renewal as well as the embryonic stem cell self-renewal. So when there is loss of 3-O-sulfur transferase, in other words, less 3-O-sulfated GAGs, there is promotion of self-renewal of both the cancer as well as the embryonic stem cell self-renewal. So this could act as a tumor suppressor, essentially, because if you have a loss, you might induce more tumor growth. And there are many processes which are altered by 3-O-sulfur transferase, but it appears that just not having enough 3-O-sulfated glycan, it significantly interferes with this extrinsic pathway of cell death, which is the FAS ligand. And when FAS ligand binds to this receptor, FAS receptor, there is activation of caspase, especially the caspase 8, which can affect both the caspases can regulate the amount of transcription factors that we discussed before, which regulate the stem cell processes, the stem cell self-renewal processes. But if caspase, is induced too much, in other words, there is very high amount of caspase, then it can even lead to cell death. But at a lower amount of caspase activation, you just see changes in the cell signaling. So you can see that these examples show you, one is that indeed the GAG, GAGs must play a very important role in the developmental processes, because essentially the developmental processes are, governed by the ability of the embryonic as well as the more adult stem cells to self-renew and grow and differentiate. Okay. So then, by extension, this molecule must have a role in regulation of these stem cells. And perhaps, most likely explanation is that because this molecule can regulate certain critical stem cell signaling, that might be one of the most important mechanisms through which GAGs might regulate the stem cells, cancer as well as the normal stem cells, growth and self-renewal. So, again, these are just the more details of some of the specific, whatever was summarized in the table. The Glycans 3, mutations can lead to the Simpson-Golabi-Behmel syndrome, where you have this dysregulated growth because of the hedgehog and wind signaling. And what happens is that the children just grow in all directions. They're very tall and their head circumference is more. And so because of this growth signaling, they're also at risk of, as you can imagine, cancer. Because, when you have dysregulated growth, you get more, there is a higher chance of cancer growth. All right, so, that's again, an example showing that proteoglycan might play a role in suppressing cancer too. Okay, so this is the EXT1 deficiency we talked about. This is the syndrome called hereditary multiple exostosis. That's where the name of this enzyme comes from, the exostosin 1 and 2 or 3 essentially, is because when there is deficiency of this enzyme, this is the type of syndrome you get. And that seems to be because of the impaired GAG synthesis, which leads to alteration in FGFR signaling, which is very important molecule early on in shaping all the organogenesis, because it can regulate the stem cell function at critical junctures. This is again that example of a thiosulfotransferase deficiency, where the death receptor signaling, as I said, because the death receptor signaling can activate or regulate the caspase, the proteases, and this proteases can degrade certain cancer stem cell or the stem cell regulating transcription factors such as NANOG and induce differentiation. So you can imagine if you remove this basal activation of caspase 3, all of a sudden, you have now more of the NANOG, and it can promote that differentiation of the embryonic stem cells and alter the stem cell self-renewal. Okay. Excuse me. So that is all the examples that we looked at were more quantitative changes in the glycosome and glycan, right? So either you remove the enzymes that are responsible for GAG synthesis, or you remove the proteoglycans which carry the GAG. But, are the finer structural details like, sulfation patterns. And we looked at a couple of the examples. Obviously, that thiosulfotransferase was one, NDSC was another one, where the specific sulfation defects can actually have a major consequences on the, on the development as well as cancer phenotype, et cetera, et cetera. This is a paper that looked at, specifically modifying the, this is specifically looking at the role of NDSC and then rescuing, that function to understand the role that the NDSC plays. In this case, it's the embryonic stem cell self-renewal, but by extension, this phenotype can be seen if the NDSC levels in the cancer cells are modulated, although that experiment hasn't been done. So essentially what this shows is that sodium chloride, which can inhibit the HS sulfation, is promoting the ES self-renewal. And you see more nano-positive cells, which these are the immunofluorescence imaging, shows that sodium chloride, you see more nano expression, and there is more growth of the colonies, suggesting that ES self-renewal can be promoted. Whereas, if you, in the NDSC deficient cells, if you provide exogenous soluble heparin, then it can rescue some of the block in differentiation that was seen. Here in the flow cytometry, you can see that there is a clear pattern change in the expression of these markers of differentiation. In the NDSC cells, when they are supplanted with heparin, you can see a clear shift in the expression of some of these differentiation markers. Essentially you can see that there is increase in collagen synthesis and GAG6 expression. That's the mRNA levels of those important molecules, which are indicative of a degree of differentiation. Okay, and also some pictures showing morphological changes, which also indicate differentiation. There's visible changes that would suggest differentiation process is activated. So basically, the rescue from, in other words, when NDST is lost, some of the differentiation function is lost, which can then be rescued by exogenous heparin. So, exogenous heparin can serve as, there must be a role. In other words, the exogenous or the soluble GAGs must be able to not only rescue the function of the endogenous GAGs when it's lost, but may also compete with endogenous GAGs in some of their functions, and thus may alter the phenotype, which is in question. Yeah. All right. So now, some of the examples that we looked at so far, even though same processes are at play in CSCs, most of those examples were specifically with embryonic stem cells and the developmental processes which are regulated by embryonic stem cells and the pluripotent stem cells. So what about the cancer stem cells? And the hypothesis is again the same, because if this GAG can regulate critical growth signaling, and then it can affect the self-renewal and differentiation like we saw in embryonic stem cells, for example, FGF signaling, as well as the hedgehog and the beta-catenin signaling. If it's altered, it plays an important, it has a major consequence on the self-renewal and differentiation of the embryonic stem cells. By extension, the cancer stem cells also use some of similar processes for the self-renewal and differentiation. So the question arises if this GAG can indeed regulate the cancer stem cell growth and self-renewal. All right. So unfortunately, that type of work hasn't been done in labs other than the ones here in VCU. So I'm just going to use examples of the work that was done in our lab, our and Dr. Desai's lab, to explain the role of these GAGs in the regulation of cancer stem cell growth. Okay, so what we did was, this figure shows that looking at one aspect of structure-phenotype relationship. In other words, and this is specifically has to do with the chain length. In other words, the length of the glycosaminoglycan molecule and its impact on CSC growth. And what you can see in this figure is that on the x-axis, you have the length, the degree of polymerization. And on the y-axis, you have the IC50, which is, indicative of the degree of growth inhibition. So the molecules which are shorter in length, up to pentasaccharides, they do not have a significant influence on the growth of CSCs. But the hexasaccharide, as you can see, which is at the nadir of this curve, has the most potent inhibition, caused the most potent inhibition of the growth of CSCs, because the IC50 is well below 50 micromolar, whereas the IC50 for the other molecule for shorter molecule was more than 300. Then for the oligosaccharides, as you increase the chain length, there is progressive decrease in the inhibition of the cancer stem cells, and then the polymers, which are the traditional GAG treatments, the low molecular weight heparin and the heparin molecules, their IC50 is well above 200 micromolar compared to less than 50 for the hexasaccharide. So even though they may partially inhibit the growth, they're just probably not the most potent sequences in inhibiting the growth of cancer stem cells. So there is a clear chain length based structure function relationship here for the heparan sulfate molecule, but also we looked at the chondroitin sulfates and the dermatan sulfates, and since for the same length, which is the hexasaccharide sequence of the chondroitin sulfate and dermatan sulfates are not as potent as heparan sulfate in inhibiting the growth. So it's not just the length, but it may also have to do with some fine structure of the glycosaminoglycan. So in other words, there are sequence-dependent phenotypic effects of this soluble glycosaminoglycan on the cancer stem cell growth. And we examined the self-renewal process too, which is shown in this bar graph, where what we do is just, and this is again using the spheroid cultures that I discussed in the previous lecture, to study the cancer stem cell growth. And essentially the spheroid growth is indicative of the stem net, especially when it's cultured, when they're propagated in subsequent cultures, so secondary and tertiary. Normally you tend to see somewhat increase in the number of spheres when you take the cells from the primary and put them in secondary and tertiary cultures. So we treated these cells only when they were in the primary culture. So from the monolayer, put them in spheroid cultures, treated them, and then after the end of the primary cultures, counted them. So the light, I would say orange bars are the effect on the primary spheroid inhibition. And you can see most of the molecules, most of these oligosaccharide sequences are causing some degree of inhibition. Obviously, the hexasaccharide is the most potent, but the most striking is that when you remove this treatment and allow those primary spheroids to be cultured into secondary and then tertiary, you can see that most of these sequences, which are not hexasaccharides, they fail to inhibit the growth after the treatment is removed. In other words, they might not be affecting the fundamental self-renewal process of the cancer stem cells. On the other hand, with the hexasaccharide, after removing the treatment, when they're subsequently cultured, there is progressive decrease. It should be actually progressive increase, in the normal condition. So this suggests that the hexasaccharides, have a major impact on not just the growth, but the self-renewal abilities of the cancer stem cells. And this was examined in many different cell lines. So what it suggests is that, there is a specific structure-based effect of these glycosaminoglycan molecules on the growth and self-renewal of the cancer stem cells. And, I'm probably not going to show you all these mechanisms in this lecture, because we have this lecture on the specifically looking at the effect of heparan sulfate on the growth signaling. We might come back to that, to examine whether this is indeed related to the GAG function of regulating the growth factor signaling or not. And so, in other words, if there is a structure-function relationship for a given target or not. Okay, so moving on. So these were some of the examples of the sulfated glycosaminoglycan and the role that they may play in the cancer and the cancer stem cell growth. Now, switching gears again, looking at the non-sulfated hyaluronan, which are quite abundant in the tumor stroma. And as I said, they actually play many different roles, and we're not going to discuss the physical properties of, abundance of the hyaluronan that plays a role in cancer growth. So we're most interested in examining its biological function that has to do with its ability to interact with proteins and regulate their function. Okay. So what has been shown is that in many different tumor types, especially pancreatic cancer and breast cancer, where there's quite rich stroma, you can find this hyaluronan molecule in quite abundance. And it's been hypothesized that the hyaluronans can promote cancer growth, one through the direct physical property, which, one was increasing the interstitial pressure. One of the ways that they can do that is through hydration, tissue hydration. And, it can affect a lot of different functions of the cells that are in that tumor stroma. They're growing in that stroma, which is rich in hyaluronan. But they also participate in cell matrix interaction, where they have a direct impact on the tumor cell growth, invasion, as well as metastasis. And this seems to be primarily, although this is not the only target, but it seems to be primarily through CD44 receptor. And what's interesting about CD44 receptor is that it's an adhesion molecule. It's a glycoprotein, and it's also a cancer stem cell marker. So high CD44 would indicate a cell has stem-like properties, essentially. But it's also a marker of migration and invasion. So the cells with high CD44 tend to be more invasive. And this seems to be one of the important targets of hyaluronane molecules. So it turns out that, and there is also sequence-dependent, the chain length-dependent interaction of hyaluronane with CD44. It turns out that in this case, it might be slightly different. It seems like, the longer chain hyaluronane tend to interact better with CD44 molecules. But essentially, by interacting with CD44 molecules, what hyaluronane does is the CD44 does not have a known ligand. It's an adhesion molecule, so this is not a classical ligand receptor interaction that is modulated by the GAGs, where they act as, they sort of regulate the ternary complex formation. This seems to be the effect of, you can say hyaluronane is almost a ligand for CD44, and then by binding to CD44, hyaluronane induces specific intracellular signaling that leads to regulation of NANOG. And if you can recall, the NANOG is a very important molecule in regulation of the stem cell phenotype. So there is a direct relationship between the hyaluronane CD44 interaction and the stem cell growth, as well as the tumor migration and invasion, because CD44 is a very important molecule for migration and invasion as well. So, there are multiple papers looking at the specific role of hyaluronane in cancer that has to deal with one of its target receptor, which is CD44. Okay. So, essentially, what I gave you is an overview of how glycosaminoglycan molecules regulate both the adult stem cells, the embryonic stem cells, and which can have a significant impact in the development of an organism, as well as the cancer stem cell growth, and how the chain length, as well as perhaps the sulfation pattern, which, I didn't give you an example here, but sulfation patterns certainly had a role in the embryonic stem cell growth, but it could also have a role in the cancer stem cell growth. Okay, so how do we use this information for translational research, or in other words, its therapeutic applications, both for regenerative medicines as well as for anti-cancer therapy development? So the stem cell therapy has been counted as one of the breakthrough treatments in regenerative medicine, because essentially, in theory, you could cause regeneration of any tissue where the tissue loss results in functional loss. So you can practically restore the function of the tissue. And if you can think of the diseases that the human beings suffer from, the most debilitating ones are the ones that have to do with the loss of tissue, whether it's the normal tissue that's replaced with the scar tissue, or because of the injury that there is there is loss of functional tissue, and all of this can be potentially repaired through the stem cell therapy. And so if you have a way of improving the stem cell therapy, then then that can be really advantageous. So if you go back to, if you recall the earlier slides, some of the things that I mentioned are the limiting factors for developing the embryonic stem cell therapy. Embryonic or the pluripotent stem cells, I'll just use them interchangeably, because essentially it's, most people would now use pluripotent stem cell. But essentially the stem cell therapy, the problems are that there is low efficiency of reprogramming. You only get 0.1 to 1% of the cells to reprogram. And that it's very difficult to maintain them because you need this feeder condition. And, you have to have a complete understanding of using various factors to get them to differentiate along specific lineage that you want them to. And because some of these factors also have, they are also oncogenes. For example, the c-Myc transcription factor of the 4 core transcription factors. It can give rise to teratoma or cancer-like growth, essentially. And so there is a potential for cancer formation too. So these are the challenges. So, if there are ways of overcoming them, and if the glycan can help in that process, then certainly would be of some advantage, right? So people have come up with this chemical approach for generation of induced pluripotent stem cells, because essentially what it is reprogramming, right? Whether you introduce transcription factors, which then regulate synthesis of various proteins, or you regulate the function of certain proteins, which also can, lead in the same direction. In other words, upregulate your OX4, NANOG, and SOX2 levels, okay? So using, library of existing compounds, people have figured out that combination of certain inhibitors can actually allow you to reprogram just using this small molecule. So in other words, you just treat the differentiated cells with the small molecules, and you can sequentially introducing the small molecules, you can achieve the induced pluripotency. And the molecules that seem to be important are the inhibitors, the activators of GSK3 beta, the ALK5, which is the TGF beta receptor, as well as the protein kinase A, and the MAP kinase signaling. So essentially, if you can, if you can devise if you can come up with a combination of various small molecules, you can achieve that pluripotency chemically. And this is one of the experiments that we conducted in our lab using Dr. Desai's molecule. You might know Dr. Desai has a library of this glycan mimetics. They're not glycans, but they mimic the function of glycan. And so we hypothesized that, if they were to mimic the function of glycans, and if the glycans were important in the growth, promotion of promotion of stemness, then we would essentially be able to achieve that sort of chemical reprogramming that we talked about. And so you can see that in cells which don't have that break, the p53 break, which I showed you earlier, it's important to remove that break p53, because even if you achieve this increased OX4, NANOG, and SOX2 levels, p53 can essentially suppress that. So once you remove the p53, then you can test and see if if your compound can increase the self-renewal. And indeed, we found that specific compounds had this self-renewal promoting ability. And when you look at their effects, essentially they affect the same pathways which are very important for chemically induced pluripotency. In other words, that ALK5, ERK MAP kinase, and the ROCK pathway. In other words, you have to use inhibitors of all this, the ALK5 separately, the ERK separately, and the ROCK separately, in order to promote that pluripotency, where you can achieve that with this just one glycosaminoglycan mimetic. Raising a hypothesis that maybe the glycosaminoglycans can actually play a very important role in this possible reprogramming, function. Okay, so that was for the embryonic or the induced pluripotent stem cell applications. What about the cancer stem cells? And again, we would look at the role of the growth signaling in this process in detail next week. But essentially, for the purposes of understanding, and the hypothesis here would be that the glycosaminoglycans can have specific properties with respect to binding to the growth factor, morphogen, or cytokine ligand and or the receptors, and that is responsible for the normal function. Normal function is, in this case, is the cancer growth. So, in other words, having certain functions, through certain functions, the glycans can promote the signaling, which can potentially be interfered with by either soluble glycosaminoglycans or the glycosaminoglycan mimetics that then can counteract the function of these endogenous glycosaminoglycans, which promote that cancer growth or self-renewal. And there are many different molecules which are being developed by different companies which regulate essentially, a variety of growth factor signaling can be modulated, including the function of the enzyme heparan. But we were mostly interested in this entity, the synthetic non-saccharide mimetics, because they have certain advantages. One, they're very simple to synthesize in a few steps. They're essentially, single chemical entities, so you're not worried about the heterogeneity that you get with GAGs. And because they're also cheap to synthesize, they can potentially have therapeutic, utility. But before we did that, before we can actually test them, I wanted to, explain you this, the tandem screening strategy that we had developed, so you guys can understand how, something like this can be adopted for high throughput screening to identify CSC inhibitors. So essentially what we did was, we know that the spheroid conditions actually promote cancer stem cell growth. So, you take the monolayer cells, grow them in the spheroid condition, you get a lot more cancer stem cells than if you just propagate them in monolayer condition. So if you can identify a molecule which inhibits growth mostly in spheroid condition, but not in the monolayer condition, that would be shown in that, that compound type C in that table, where you have essentially no effect on monolayer growth, but a strong effect on spheroid growth. Then you have something that could target either the progenitors or the stem cells. Now, how do you distinguish between those two? So what we did was, after this step, you treat these cells at this level, the primary spheroids or the primary monolayer culture, and then remove the treatment, and then subsequently propagate the spheroids into secondary and tertiary without any further treatment. Now, if your molecule was only targeting the progenitors, the C molecules, the type C1 molecules would, retain that growth inhibition even when the molecules are removed, because essentially what you had impacted is the self-renewal ability of the stem cell. So you take that stem cell, propagate in the secondary and tertiary conditions, they won't form more spheroids because their self-renewal ability has been impacted. On the other hand, if you only inhibited the progenitors, the stem cells have escaped that growth inhibition. Then in the secondary and tertiary cultures, the stem cells would be happy and they would just make more and more spheroids. So essentially, you've lost that inhibitory potential. So by using these two screens in tandem, we can actually identify molecules which can selectively inhibit the cancer stem cell self-renewal. And indeed, using over, I think this is 53 compounds, but by now we have screened about more than 75 compounds. And what we observed was that we indeed found the C-type molecules, which are, these molecules, which are 11 molecules, which inhibited growth in the spheroid condition, but not so much in the monolayer condition. And these were of, further interest. So when we stopped the treatment and then cultured those spheroids treated with these 11 molecules and observed their retention of inhibition of the spheroid growth, only 3 of these molecules actually inhibited the growth in the secondary and tertiary conditions, suggesting that they might have true ability to inhibit the CSC self-renewal. And what was really interesting was that this molecule that we eventually selected to study further is a molecule which is a dimer of this quercetin, sulfated quercetin, which had absolutely no effect. But interestingly, the same quercetin, when it was in a trimer formation, it actually only inhibited the progenitors and not so much the CSC self-renewal. So there is a clear relationship between the length of this sulfated sort of a GAG mimetic, similar to what we had observed before with the natural GAG, essentially. And so that actually, led us to believe that there might be some, very interesting biological phenomena that we might have come across using this tandem screening strategy. And we found that this molecule was inhibiting CSC growth across many different cell lines, irrespective of what the underlying mutation profile of the cell is. So essentially speaking, it's a broad inhibitor of cancer stem cell because it must be inhibiting some fundamental processes which are critical to the cancer stem cell growth. And these are the in vivo effect. Just to show the effect on CSCs, is that the IP injection in a mouse that bears a xenograft, after the treatment, you remove the xenograft and analyze it for certain, for the cells that express certain CSC markers or the cancer stem cell markers. You can see a robust downregulation in the animals that were treated with this compound. But interestingly, even though it inhibits the growth in the cancer stem cell growth, it had very little impact on the normal stem cells because we cultured both the bone marrow cells into the ex vivo cultures as well as the intestinal stem cells into what we call organoid culture, where you can take the intestinal crypts and grow them, and actually they make small intestines ex vivo on a special matrigel that's supplanted with certain growth factors. So it had no impact on that organoid formation, even though it strongly inhibited the cancer stem cell growth. So that's what's really interesting, is that you could identify these are the glycosaminoglycan mimetics, but potentially natural GAG sequences which may have a very selective effect on the cancer, but not the normal stem cells, and that could have a very fundamental impact on the development of anti-cancer therapies. And so what we actually found was that this G2.2, which was the non-saccharide mimetic, is a structure. Well, using the in silico molecular dynamics method, we showed that this molecule could be structurally very similar to hexasaccharide. And then in the other studies, we actually showed that functionally this molecule behaves just like the natural hexasaccharide sequence, which I showed you before, was the most potent sequence of all the natural heparan and the chondroitin sulfate and the dermatan sulfate sequences that we had examined, was the most potent inhibitor of cancer stem cell growth and self-renewal, importantly self-renewal, because there is a progressive inhibition of growth despite removing either the hexasaccharide treatment or this G2.2 treatment. And that can only happen if you fundamentally affect the self-renewal processes. So I'm not going to go into the details of the mechanisms and things like that. Some of that we would hit upon when we deal with the interaction of the sulfated glycosaminoglycans with the growth factors and things like that. So finally, one more use of this is essentially the non-sulfated glycan. I showed you that the hyaluronans have very specific targets. One of them is the CD44 receptor, which is found mostly on cancer cells, which have the CSC-like property, or the ones that probably are going to behave badly because they have more invasive property. So in order to target those cells specifically, what one could do is actually use hyaluronan to deliver as a Trojan horse to deliver the cargo. And people have started using hyaluronan in their nanoparticle preparation to deliver certain anti-cancer drugs to the CD44 expressing cells. And that could also be a potential use of these glycosaminoglycan sequences in developing novel therapies against cancer. So in conclusion, what we can summarize is that so far, based on specific examples that we looked at, it seems like glycosaminoglycans play a very important role in the normal stem cell growth and fate determination, which is essentially, when the normal stem cell growth is affected, their differentiation along a certain lineage. The one of the three important lineages, the endoderm, epiderm, and the mesoderm, can be significantly affected, and that could result in altered growth as well as organogenesis, and that could lead to developmental defects. Also, the glycosaminoglycans, we looked at the very specific role, both the quantitative depletion of glycosaminoglycan as well as alteration in the sulfation pattern also affected the growth of embryonic stem cells and/or induced pluripotent stem cells. And then finally, we can use the glycans or their mimetics as a potential agents to both as chemical biology tools to understand the role of GAGs in the cell signaling as well as the CSC regulation, and then perhaps develop them as potential anti-cancer therapies, which selectively target cancer stem cells while sparing the normal stem cells. And those treatments are very hard to come by because nature doesn't have many molecules which have that ability to distinguish between the cancer and the normal. And potentially with the GAGs, we might have, in GAGs, we potentially might have that function, which is very important to, tap in. Okay. All right. So I think that was my last slide. If you have any questions, we can address them now.
That was good. Exactly 450. So, any questions? Okay, none from here. Anybody from people logged on?
I think we're good, thanks.
Okay. Very good. Thanks, Bhaumik. That was a nice talk and greatly appreciated.